Any consideration of sugar metabolism must include discussion of the ways in which sugar is stored and released in the body. In animals, this is glycogen and will be the subject of this talk. In the first part of the talk, what I will do is go through a bit of the structural and enzymatic considerations in the breaking and making of glycogen. And in the last part of the talk, I'll discuss glycogen from a regulatory point and how that regulation helps the animal to meet its needs. Now, glycogen is a polymer of glucose, as I've noted in another one of the presentations. It is actually a very large polymer of glucose, as you can see on the screen on the right. Glycogen is stored in animals in liver and muscles. In liver, it is used to provide a constant supply of glucose to the bloodstream as the animal needs it. And in muscles, it's an immediate source of glucose for the contracting actions that muscles have to take. Now, glycogen is similar to amylopectin of plants and differs only in the amount of branching that the molecule has. Glycogen has branches about every 10 glucose residues, whereas amylopectin has branches about every 50 or so. Glycogen helps to modulate blood glucose, and it is because it is that battery or that storage form of glucose. The liver can tap it as necessary to release blood glucose into the blood and allow that blood glucose level to stay as needed. Now, glycogen in its synthesis does not start with a glucose. It actually starts with a protein called glycogenin, and to that protein is attached the first glucose molecule. And the attachment of that first glucose molecule leads to the structure that you see here. And as you can see, this structure can be quite complex with up to 20,000 glucose units within a given glycogen entity. Now, this differs a bit from the plant storage glucose forms, which you'll have on the order of about 400 or so. Now, the metabolism of glycogen turns out to be relatively simple compared to other metabolic pathways that I've talked about in these presentations. The complexity of glycogen actually relates to its regulation. We see on the screen a depiction of the way in which glycogen is broken down. So glycogen is broken down by an enzyme called glycogen phosphorylase. And in many places, this is abbreviated and simply called phosphorylase. As we will see, there are a couple of different forms of phosphorylase for consideration. Now, glycogen phosphorylase is an interesting enzyme in the way that it breaks glycogen down. Many molecules, like sugars and so forth, are broken down by hydrolysis using water to break bonds. Glycogen phosphorylase does not use water. Instead, it uses phosphate, as you can see here. And so this is, instead of being called a hydrolysis, is called a phosphorylasis. Now, that's not just an important thing of nomenclature, but rather it actually is saving the cell energy, as we shall see. The phosphorylasis of glycogen results in production of a molecule called glucose 1-phosphate that you can see on the lower left, and a glycogen that's been reduced by one residue. So what has happened in this catalytic action is that the enzyme has broken the bond, the 1,4 bond, between the end of a glycogen chain. Now, it's important to recognize that glycogen phosphorylase works only on ends. It doesn't cut in the middle, it works on ends, and it sequentially chews its way into a glycogen. So one of the reasons that the branchedness, as it were, of glycogen is important is because that the more ends there are, the more glycogen phosphorylases can start on ends and sequentially release a tremendous amount of glucose very quickly. We hear stories about somebody who's in a terrifying situation. They do incredible feats of lifting something heavy that would seem otherwise impossible. And the ways in which this actually happens because of the branchedness of glycogen, which releases this burst of glucose that people use to accomplish what they do. Glucose 1-phosphate is readily turned into glucose 6-phosphate by the enzyme phosphoglucomutase. Now, this is a reversible reaction, as you can see. The significance of the reaction, though, is that glucose 6-phosphate is produced. And if you remember, we started with a glycogen that had glucoses that had no phosphates on them. We have at this point an intermediate in glycolysis, glucose 6-phosphate, that does have a phosphate on there. And that phosphate got on there not by the use of ATP. If you recall from the glycolysis presentations, hexokinase uses an ATP to put a phosphate onto glucose to make glucose 6-phosphate. Here, this phosphate got put onto the glucose from the glycogen by using a phosphorylasis reaction that did not require ATP. This saves the cell energy. And the energy for putting that phosphate on actually came from the breaking of the 1,4 bond by the glycogen phosphorylase. So cells are very efficient. Cells use whatever they have available to them to do things as efficiently as possible, and this is a very good example. Now, glycogen breakdown proceeds, as I said, by the action of glycogen phosphorylase working from ends. And I also noted that glycogen is a very branched molecule. We can see on the screen here a branch of glycogen and learn a little bit about the way that glycogen phosphorylase works. You see on the top the branch, and you see the end of the molecule where we have 10 different blue residues that are shown on the left. Those 10 residues are all targets for glycogen phosphorylase. So as the enzyme acts, it goes through and it releases those 10 sequentially in the reaction that I showed at the beginning of this lecture. It takes 10 phosphates, and 10 glucose 1-phosphates are produced as a result. Now, at that point, glycogen phosphorylase won't go any further. So glycogen phosphorylase has a limit in how close to a branch it will work. You note that the branch is at the base of the yellow molecules shown on the very top. Now, as we move down to the second part, you see we've changed the color of those glucoses so that 3 of them are still in yellow, and 1 is now shown in the sort of orange color, and there's a reason for that. There's an enzyme called debranching enzyme that plays the second role in breaking down glycogen. Debranching enzyme takes the 3 yellow residues that you see in the square and transfers them down to the chain below. So because glycogen phosphorylase can only work within about 4 units of a branch, this debranching enzyme plays the important role of rearranging the molecule so the glycogen phosphorylase will have additional substrates to work on. Debranching enzyme is also interesting in another respect, and that is it actually catalyzes 2 different reactions. The first reaction you can see on the screen, which is the transfer of the 3 residues of glucose down to the lower chain. What's left behind is that single molecule that's shown in the above the main chain. That single molecule has that alpha 1,6 bond that's essential for that's part of the glycogen branching. That last molecule is removed by a hydrolysis reaction. So water is actually used to release that, and glycogen debranching enzyme catalyzes that reaction. So it's a very flexible, it's a very remarkable enzyme in doing what it does, but because of that, you can see that an unbranched form is now left behind, and glycogen phosphorylase can come back in and continue the phosphorylasis that it was doing originally. I should also note one other point here, and that is that the release here of glucose is the only place in glycogen metabolism where free glucose is released. All other releases are glucose 1-phosphate. So what you've seen so far is all the reactions involved in the breakdown of glycogen. It's a very simple pathway, and as we will see, the synthesis of glycogen is almost as simple. In the synthesis of glycogen, we start with glucose 1-phosphate. So where would glucose 1-phosphate come from originally? Well, imagine that you had a cell that had gotten a bunch of glucose into it. The cell could use hexokinase in the glycolysis pathway to convert glucose into glucose 6-phosphate. And then phosphoglucomutase, which catalyzes the reversible reaction I showed earlier, can convert glucose 6-phosphate back to glucose 1-phosphate, as you see here. Now, that turns out to be pretty important because cells, liver cells in particular, will release glucose. They will also take up glucose after a meal, for example, when the glucose concentrations are high. Dealing with that glucose is important, and storing it as glycogen, as we see here, is essential for them to do. So that phosphoglucomutase plays a dual role both in the breakdown and the synthesis of glycogen. Glucose 1-phosphate, in the first step of glycogen synthesis, combines with a molecule called UTP. Now, UTP is, of course, a ribonucleotide that's used in the making of RNA. In this case, it's used to make a molecule that can be used to build glycogen. Now, the enzyme that catalyzes this reaction is UDP glucose pyrophosphorylase, as you can see. And what happens in this reaction is that a pyrophosphate, the PPI that you can see in the lower part of the figure on the lower right, is released, and the remaining part, the UDP, is attached to the glucose molecule. Now that bond between the glucose molecule and the UDP is a high energy bond. It's an example of what I like to call an activated intermediate. Now, activated intermediates have a very important property. First of all, they have high energy bonds, and the high energy bonds are used to transfer a part of themselves to something else. Now, that's actually what we see in this image on the screen. UDP glucose is getting ready to use its energy to transfer a part of itself to glycogen. And the part of itself it's transferring, of course, is glucose. So this is a reaction that's catalyzed by glycogen synthase, as you can see. And it's a fairly simple reaction. What the glycogen synthase is doing is grabbing that glucose and making a 1,4 bond so that the glycogen chain is growing by one more glucose. The remaining product, UDP, can then go back and be phosphorylated and used for making RNA or making additional UDP glucoses. Now, of course, all that glycogen synthase is going to do is make glycogen with alpha 1,4 bonds. And you recall that glycogen has many branches. So then the question is, how do the branches get there? And the branches get there by an enzyme that has a name that's very long that most people call branching enzyme, and so I've decided to call it branching enzyme here. You can see that what branching enzyme does is it grabs a section of a long alpha 1,4 polymer of glucose on a glycogen chain and then clips it and transfers part of it backwards to where it can make an alpha 1,6 bond with another sugar. And it's in this way that the branches actually get into glycogen. So you've seen really all there is to see with respect to glycogen synthesis and glycogen breakdown. There's only a handful of enzymes that are involved in those entire pathways. Now, that allows for some simplicity in terms of regulation, but as we will see, that regulation has a bit of complexity, but it's focused completely on the enzymes involved in the direct synthesis and the direct breakdown. These, of course, are the glycogen phosphorylase and the glycogen synthase. Now, the body works with glycogen metabolism largely through hormonal action. And hormones are involved in transferring information in one part of the body to another. So, for example, a muscle cell that is undergoing a lot of energy burning from contracting and so forth, can release a signal that says, hey, I'm needing some glucose, and that signal can travel to the liver, for example, and then cause a process to release glucose. The release of hormones is an organismal function. It's important to remember that as we talk about some of the pathways that I'll be talking about here for hormone stimulation. And the reason that that's important is because the needs of the body are quite varied. Muscles have a different need than liver has. Liver is there to supply, muscles are there to use glucose, for example. So what we're focusing on here are largely liver cells. Liver cells supplying the glucose, or in some cases storing the glucose for the body. The regulation of glycogen metabolism occurs through one of the hormones that you see here. Now, there are a couple of hormones that play a role in telling the liver, hey, the body is needing some glucose. These include epinephrine and a peptide hormone called glucagon. Epinephrine is also known as adrenaline, for example. The binding of either one of these hormones to the receptor in the cell membrane that you see on the upper left causes a change in the receptor, a slight change in structure of the receptor, and that change is communicated inside of the receptor to the inside of the cell. Now that's a very critical step in this process because what's called signal transduction that I'm talking about here is an essential way that the outside part of the world connects with the inside part of the world of a cell. In the signal transduction, the inside of the receptor, and the receptor here that I'm talking about is called the beta-adrenergic receptor. The beta-adrenergic receptor interacts with a protein called a G protein, and you see that below in orange. That's labeled with an alpha, a beta, and a gamma. That protein has 3 subunits, and the functional part of that protein, for our purposes, is the part that has the nucleotide GDP located in it, and it's the GDP that actually gives the protein the name of a G protein. It means it contains guanosine nucleotides. The action of the binding of the hormone that changes the receptor changes the interaction between the receptor and the G protein. And that causes two things to happen. One thing that happens is that GDP is released from the alpha subunit and replaced by GTP. Second, the interaction between the alpha subunit and the beta gamma subunit is interrupted. So the beta gamma subunit is released, and the release of the beta gamma subunit allows the G protein to travel from the receptor over to a membrane-bound enzyme called adenylate cyclase. Adenylate cyclase is an enzyme, as its name suggests, and what it does is it catalyzes the conversion of ATP into a molecule called cyclic AMP. Now, cyclic AMP is an example of what we call a signaling molecule, and cyclic AMP is involved in many signaling pathways. We're looking at one of the pathways right here. Well, the reason cyclic AMP is important is it is an allosteric effector on protein kinase A. Now, as we will see in this presentation and subsequent presentations, protein kinase A is central to phosphorylation of many proteins, and those proteins have significant changes that happen in their activity as a result of that phosphorylation. So the addition of cyclic AMP to protein kinase A converts it from the inactive form on the right to the active form on the left. That active form on the left then is able to start phosphorylating proteins. In the inactive form, it's not able to do that. So in the absence of cyclic AMP, protein kinase A will not phosphorylate anything. The phosphorylation by protein kinase A, I want to focus on the arrows moving down first, and then I'll talk about the one moving to the left later. Protein kinase A phosphorylates proteins, that's how it gets its name, and the phosphorylation that it does first is on an enzyme called phosphorylase kinase. Again, whenever you see the name kinase, you should think puts phosphate onto. And so what phosphorylase kinase does is when it's activated, it puts a phosphate onto another enzyme. Phosphorylase kinase is converted from the inactive form on the right to the active form on the left by that phosphorylation, and that active phosphorylase kinase converts glycogen phosphorylase B, which is relatively inactive, into the glycogen phosphorylase A, which is very active. And so what the glycogen phosphorylase A does is it goes and catalyzes the reactions that we've already seen. That is the breakdown of glycogen to make glucose 1-phosphate that can then be used later for energy. A second thing that protein kinase A phosphorylates is it phosphorylates the enzyme glycogen synthase that we've already seen. Now, glycogen synthase, of course, is involved in the synthesis of glycogen. The effect of phosphorylating glycogen synthase is to do exactly the opposite thing. In the case of glycogen synthase, the B form is the active form, and the A form, which is phosphorylated, is inactive. As a result of this, glycogen synthase is converted into an inactive form. So what's happened here is what we call reciprocal regulation. Protein kinase A has started what we call a phosphorylation cascade that has resulted in the different effects on catabolic and anabolic pathways. The catabolic pathway glycogen breakdown is activated, and the catabolic synthetic pathway is inhibited. So as we've seen, this system is designed then to put phosphate onto glycogen enzymes, and these addition of phosphate glycogen on the glycogen enzymes is activated by the hormone stimulation, and you've seen a little bit of the effects that these can have on the activity. Glycogen synthase, the synthetic enzyme, is less active with phosphate, and glycogen phosphorylase that breaks down glycogen is more active with phosphate. Now, this reciprocal regulation is very, very important for these metabolic pathways, and reciprocal regulation as a principle is very important for catabolic and anabolic pathways in general. Now, you can see here the phosphorylation that's happened on there, and the phosphorylation or the addition of those phosphates must be removed in some way because cells are control freaks, as I like to say. Cells, if they want to do something, they want to also be able to reverse what they do. And they have to be able to reverse what they do because it's really important here. Glycogen is a resource of the body, and the activity of a glycogen breakdown enzyme is such that it could very readily break down the glycogen and waste all the glycogen if it was doing that when the glycogen was not needed. So it's important that this breakdown be controlled, and the breakdown is controlled by removal of phosphates, as we should see by an enzyme called phosphatase. Now, another interesting consideration occurs at the top. So when we're talking about reversing and stopping this pathway, if we stop the lower part of the pathway, but we don't stop the upper part of the pathway, then this signaling will continue to activate glycogen breakdown. So it's important that these internal things inside the cell be turned off at all levels. And this is a very interesting process. The G protein that you see on the left is, with the GTP bound to it, the G protein is a very inefficient enzyme. And because it's an inefficient enzyme, it is able to do what it does. Now that may seem very odd, but it's true. What happens is the G protein, besides activating adenylate cyclase, has an enzymatic activity of its own. And the enzymatic activity of its own is that it cleaves GTP and leaves behind GDP. Well, when the G protein has GDP on it, we've already seen what happens to it. It goes back to the adenylate cyclase, it goes back to the receptor and binds to the receptor, and the beta and the gamma bind to it. So the G protein has a way of turning itself off. But what's important is because it's an inefficient enzyme, it doesn't cleave GTP too fast. So by having an inefficient enzyme, it takes a few seconds to a few minutes for that conversion to happen so that it's able to communicate the signal to the adenylate cyclase to be active, but it is able to turn itself off automatically by that. Now, the other molecule that's interesting in this respect is cyclic AMP, because cyclic AMP must be dealt with, otherwise it will continue to activate protein kinase A. And that's what happens, as we can see here in the next slide. Cyclic AMP is broken down to AMP by the enzyme phosphodiesterase. And when that happens, AMP will not stimulate the process, as I've talked about, and so the signal has been turned off. Now, you'll note above that that enzyme phosphodiesterase is inhibited by caffeine. Now, caffeine has numerous effects in the body, but I always like to remind people that you think about that buzz you get from caffeine. What's happening with that buzz? Well, part of that buzz comes from the fact that the phosphodiesterase is being inactivated. And when the phosphodiesterase is being inactivated, protein kinase A is being activated, and glycogen breakdown is being stimulated. So what you're doing by drinking caffeine, at least to a limited extent, is increasing your blood glucose. That's part of your buzz. Now, so in summary here, when glucose levels are low, the body signals the liver with epinephrine or glucagon, and it increases the glucose concentration by favoring glycogen breakdown and inhibiting glycogen synthesis, the reciprocal regulation that I've talked about before. Now I want to spend a little bit of time talking about how we reverse all the process. So a reversal of the process happens with another hormone, and that other hormone is one that we've all heard about, and that's insulin. Insulin, as we will see, has the effect of countering the entire epinephrine-glucagon system. Now, insulin stimulates an enzyme called phosphatase that I mentioned earlier, and what phosphatase does is it removes phosphates from the glycogen enzymes. This favors glycogen synthesis because it was the addition of phosphate to the glycogen synthase that inactivated it, so removing the phosphate will activate it. Conversely, we take the phosphate off of glycogen phosphorylase, and we will have the effect of reducing its activity. Insulin also stimulates the uptake of glucose by cells, and this is important because blood glucose can be hazardous for the body. So getting glucose out of the blood and into the cells is important. In this figure, we can see a little bit of the results of insulin action on cells. We see in yellow that insulin binds to a receptor of its own called the insulin receptor. That binding to the insulin receptor stimulates several things to happen. Now, as I noted, the liver cells, one of the things that happens is that they are stimulated to take up glucose from the bloodstream. The body treats glucose like it's a poison, and in some ways it may well be a poison because people who have trouble maintaining their blood glucose levels have many problems. We know, for example, the issues associated with diabetes. So maintaining proper glucose levels is important, and insulin plays a role in that process. We see insulin stimulates the movement of a protein called glucose transporter 4 to the cell surface. And so that green dimer that you see on the cell surface has moved there as a result of insulin action. The glucose is brought into the cell when that happens, and you can see the glucose concentration increasing. A couple of things can happen with that. Glucose in the cell, again, the cell is now going to treat it like it's a poison. The cell deals with that glucose in two ways. One is it reduces the free glucose concentration by making glycogen. That takes glucose away out of the free system and makes glycogen, and glycogen is not a problem. In addition, glycolysis is favored by the breakdown of glucose, so that also has the effect of reducing the glucose concentration. Now, the byproduct of glycolysis and breaking down glucose is you make a lot of pyruvate. If you're not getting plenty of exercise, what happens is a lot of pyruvate gets converted into acetyl-CoA, and acetyl-CoA goes to fatty acids, and you can probably figure out where that's headed. So, when glucose levels are high, insulin decreases glucose concentration by favoring glycogen synthesis, as I noted, and by inhibiting glycogen breakdown. So, when glucose is needed, epinephrine does its job and stimulates the release of glucose from glycogen, and when glycogen is abundant, insulin stimulates the dephosphorylation of those same enzymes to use the glucose in either glycogen or glycolysis. So maintaining sugar balance in the body is an essential function. And in this lecture, we've seen how glycogen metabolism is managed to help the body to manage its own glucose needs. With these managed needs done properly, the body functions having glucose when necessary and being able to store glucose when there's an excess.